All right, guys, today we're going to talk about protein synthesis. Protein synthesis is how we go from the instructions of how to make you what you are to the actual proteins that build your body. There is a PowerPoint that goes along with this that you need to watch. You need to, read through all of the slides, watch the 4 or 5 embedded videos within there because that's going to give you the kind of reading the details, the minutia that I'm going to talk through on the screen right now. Pay attention to these different steps. Step number 1 is transcription. We need to rewrite the message. Now remember, your DNA is found up here in your nucleus, and it can't leave the nucleus. That poses a problem because your proteins are made in the cytoplasm on the ribosomes. So somehow we have to get the instructions from the nucleus to the ribosome. So up in here we have your DNA, and it's hanging out in the nucleus. Let's say you had breakfast this morning and you had milk with your breakfast. Remember when we talked about biochem? You now need an enzyme called lactase to break down the milk that you drank. So there is an enzyme called RNA polymerase in the nucleus that scans your DNA to find the gene for how to make lactase. When it finds it, it looks for a promoter region. This is like the flag that says, hey, here's where that gene is. When RNA polymerase finds the promoter region, it opens up a bubble of your DNA just where that gene is. And I'm just going to throw some random nucleotides in here so we have something to play with. Remember, on our DNA, A always bonds to T, C always bonds to G. Here we go. I've opened it up. Here's where the gene is. RNA polymerase says this is what you need to transcribe into a new message. And so we're going to make something called messenger RNA. So the messenger RNA is going to take the instructions from the nucleus to the ribosome. When I bond, T goes to A, but now because I'm making RNA, A goes to U, G goes to C, C goes to G, G goes to C, C goes to G. Couple things we need to know about RNA. Markers have not been used all year. So RNA, single strand, A bonds to U instead of A to T. No T's in RNA. And we have ribose as the sugar. Those are the 3 differences between DNA and RNA. Okay, so now I've got this transcribed. I have it rewritten as mRNA. mRNA leaves the nucleus through a nuclear pore and goes to a ribosome. On the ribosome, it is read in a group of 3 called a codon. This is the code for what amino acid we are going to put in what order. So the codon is the code. It's the secret message. The codon is read and matched up with the anticodon. Anti is the opposite, right? So where there's a G, I get a C. Where there's a C, I get a G. Where there's a G, I get a C on the anticodon. C goes to G, U goes to A goes to U. Because remember, I'm an RNA, so no T's. Attached to your anticodon is an amino acid. The amino acids would rather hang out with their own kind than hang out with this anticodon. And so when they're put next to each other, they form a bond called a peptide bond. It's a covalent bond that's found in proteins. Talked about it back in biochem. And once it forms the bond with its neighboring amino acid, it breaks off of its tRNA, and the tRNA goes out and finds another amino acid. Now we have our protein. This growing amino acid chain becomes our protein. A couple of things to note. Once the mRNA is translated, right? So transcription, you rewrite it, translate it, we change it into the language of amino acids. Once the mRNA is translated, it gets broken apart. So it's a one use and done. When we're talking about these new vaccines, these mRNA vaccines that are out, Pfizer and Moderna, these are mRNA vaccines. Never does this mRNA go into the nucleus. This mRNA is not long lasting. It basically goes into your body in these fat bubbles and gets absorbed into the cell, and the cell's ribosomes translate that mRNA into a protein chain. That protein chain is the spike that's found on coronavirus, just the spike. Not the information on how to make you sick, not the information on how to make a complete virus, just the itty bitty spike that then gets recognized by your immune system to build up a complete immune response to then give you the antibodies that will give you immunity so that if you come in contact with the entire virus, you won't get sick again. All right, one last thing that I want to show you is, well, if I have my codons, how do I figure out what the amino acids are that go on them? So I just Googled amino acid chart, right? And I get one. When you look at this amino acid chart, you read from the inside out. So if my first codon is GCG, I would go GCG and I get alanine. If my second codon is CUA, I'd read from the inside out, CUA, and get leucine. All right, make sure you guys watch your PowerPoint. Make sure that you watch the embedded videos. Make sure that you're able to replicate what I just did, that you could draw this and explain transcription going from DNA to mRNA, the messenger RNA, and leaving the nucleus, and translation going from mRNA to an amino acid chain or a protein.